Fungi are heterotrophs, and they absorb nutrients from outside their bodies. Unlike plants, which are photoautotrophs, they do not have photosynthetic organelles, but they do have diverse and different type of enzymes to exhibit diverse life cycles, such as decomposers, parasitic, and mutualistic. We'll be learning the morphology and the common body structures of fungi. The most common body structures are multicellular and single cells, called yeasts. So fungi have hyphae, chitin, and mycelium. Hyphae are the little tiny strings, and the individual strings are called hyphae. And interwoven and the network of hyphae is called mycelium. Chitin is used on the hyphae cell walls. It reinforces the cell walls because it prevents the cell walls from bursting. Remember that they live in a hypertonic. Well, they are hypertonic because they're absorbing nutrients 24/7. And this prevents water from balancing these things out and causing them to burst. They want to absorb nutrients as much as possible. Fungi will also spend more time and energy creating longer hyphae than making them thicker. This creates a high surface area to volume ratio. There's two types of hyphae. Hyphae that are separated by that are divided into cells by septa. So this is a septate hypha, and hyphae that do not. They have one continuous long cytoplasm, which is called a coenocytic fungi or fungi. So there's specialized hyphae called mycorrhizal fungi. And these guys have arbuscules. And arbuscules penetrate cell walls, or unlike ectomycorrhizal fungi, they penetrate cell walls, and it's a mutualistic relationship between the plant and the fungus. The fungus will provide the plant nutrients and minerals, while the plants will give them sugars, which they produce through photosynthesis. There's two types of mycorrhizal fungi. There's arbuscular mycorrhizal fungi and ectomycorrhizal fungi. Arbuscular would penetrate their arbuscules into the cell walls of plants. So they will penetrate the plant cells, while ectomycorrhizal fungi would not. They will rather form around the cell rather than penetrating it, and will sometimes grow their hyphae in between plant cell walls. So the life cycle of fungi is pretty simple. Spores are haploid cells called there's cells haploid cells called spores, and there's their way of reproducing, and they colonize soils by the dispersal of these spores. They can either propagate themselves either sexually or asexually. So here is a very simplified version of fungi reproducing. So here it is plasmogamy. Plasmogamy is the fusion of the cytoplasm. The eukaryotic stage, or the dikaryotic stage, is when two different nuclei are in the same cytoplasm but have not yet fused. Karyogamy is where the both nuclei fuse and become a zygote. The diploid state is short-lived in fungi, and it immediately goes through meiosis, creating more spores germinate. It either goes through asexual reproduction or sexual reproduction. Fungi nuclei are normally haploid, but there's an exception, like the zygote. And sexual reproduction requires the fusion of hyphae from different mating types. The reason why there's mating types is that it prevents a fungus from mating with itself or with its similar friends or cousins or brothers or sisters. This creates genetic diversity because it allows a fungus to mate with different types of other funguses. Fungi, fungi, fungi uses sexual signaling molecules called pheromones. We also use pheromones to communicate their mating type. We just went over these. We'll be going over asexual reproduction. In addition to sexual reproduction, many fungi can reproduce asexually. Molds produce haploid spores by mitosis and form visible mycelia. So a yeast are single cells. Instead of producing spores, yeast reproduce asexually by simple cell division and pinching of bud cells from a parent cell. So one parent cell gives a rise to many other cells. And that's pretty much how yeast reproduce. So funguses are more related to animals than to other plants and eukaryotes. So when you see a mushroom, you are genetically closer to the mushroom than to a plant. Fungi were the earliest colonizers of land, and they are basically their ancestors were aquatic. There's fossil evidence that indicates fungi formed mutualistic relationships in early land plants, but the twist is that early land plants did not have roots, which mycorrhizal formations could form. But it was later realized in a genomic analysis that there's only 3 genes involved in mycorrhizal formation, with or without roots. The fungi diversity. There's currently about 100 species of fungi that are recognized. Some estimate the actual number of fungal species may be 2.2 to 3.8 million. So here we have like a Cheeto puff looking fungi. Here we have like strawberry jelly, foam, whatever it is. It looks really cool. I want to eat this. Probably would be bad for me. And here we have tortillas. We'll be exploring individually a few of these fungi, starting with the Chytridiomycota. Chytrids are found in terrestrial, freshwater, and marine habitats, also hydrothermal vents. They are decomposers, parasites, and mutualists. Chytrids are unique among fungi because they have flagellated spores called zoospores, like sperm, which are flagellated, but spores, meaning that they can swim. Another is mucoromycetes, or actually Mucoromycota. This includes fast-growing molds, parasites, and commensal symbionts. An example of this is the black bread mold, and we'll be exploring its life cycle here. In asexual reproduction, asexual sporangia produce genetically identical haploid spores, while in sexual sporangia produce genetically diverse haploid spores. So here we start with 2 mating types of different hyphae. Mating type negative and mating type positive. Once they come in contact, they will form a gametangia, meaning that these are close together. They're like a couple, and they're like holding hands, basically. Plasmogamy, meaning that they fuse, both cytoplasms of the different mating types fuse, and all 3, or actually all total of 6 nuclei are in the same one cytoplasm. This is called a young zygosporangium. Later, it will become a zygosporangium, and they will undergo karyogamy, which, of course, karyogamy is the fusing of these nuclei with their pairs. So these 2 will fuse, these 2 will fuse, and these 2 will fuse, forming 3 diploid nuclei. And remember that the diploid stage of funguses are short-lived. A cool fact about the zygosporangium is that they form a thick coat around it. So zygosporangium can remain viable and can remain dormant for several months. And then after that, the diploid nuclei will undergo meiosis. A stalk will rise out the zygosporangium, and a sporangium will be on top. It looks like a little branching, like something off like Aliens or something. Alien versus Predator, I think. The sporangium will either go asexual or sexual reproduction, and it will spit out spores via mitosis. Asexual spores are genetically identical, while the spores in sexual reproduction are different. 80% of plants share a mycorrhizal relationship with glomeromycetes. Some random fact. Ascomycetes, sac fungi, live in marine, freshwater, and terrestrial habitats. This includes plant pathogens, decomposers, and symbionts. Ascomycetes produce sexual spores in sac-like acid. Most develop fruiting bodies called ascocarps. But during asexual reproduction, a unique trait of ascomycetes are asexual spores called conidia. So conidia are here, so they're specialized hyphae, and it's only found in asexual reproduction of the ascomycetes. So here is the typical ascomycete life cycle. We'll start with asexual reproduction. So here we have the hyphae, and the hyphae would have specialized hyphae called a conidiophore. These conidiophores will spit out their conidia, or equivalent to a spore, and it will either go through asexual reproduction, or sometimes the conidia can also mate with a different mating type. Once that happens, plasmogamy occurs now we're talking about asexual, since asexual reproduction is pretty simple. It's a short circle. In sexual reproduction, the conidia fuses with the mating type of another ascomycetes, and they form an ascus. An ascus is dikaryotic, and it also has dikaryotic hyphae. So remember, this is a karyotic stage, meaning that the two nuclei haven't fused yet until karyogamy. Once karyogamy takes place, those two nuclei fuse and become a diploid nucleus, which is a zygote. And it's short-lived, of course, because it's a fungus, and it will undergo meiosis. It will go one round of meiosis, which is 4 haploid nuclei, and those 4 haploid nucleuses will undergo mitosis, creating 8 ascospores. And there's more of these ascospores, and here is the ascocarp, which is the fruiting body, and it will spit out these ascospores, and these ascospores can either go through asexual reproduction via the conidiophore, or they can become mycelia, and those mycelia can either mate with their mating type. So here is a closer look on asexual reproduction. The spores will germinate, the hyphae will grow, and if it wants to go through asexual reproduction, it will grow and grow and grow until the conidiophores, a specialized part of hyphae, release their conidia, their mating type is negative, and they can germinate themselves and become mycelium again, and they can germinate themselves and grow into hyphae, into mycelium, and so on. You get the idea. It's pretty simple. This is what I explained up there. The canidia can either go through asexual or sexual reproduction, but now we're talking about sexual reproduction. So the canidia will fuse with its mating type, undergo plasmogamy, form an ascus, which is they're in the same cytoplasm, but not yet fused. Karyogamy, meaning they now fuse. The diploid zygote is short-lived. It will undergo one round of meiosis, creating 4 haploid nuclei, and a few rounds of mitosis, creating 8 ascospores. And these ascospores will be spit out by the ascocarp, be dispersed through the environment, and germinate, and grow into mycelia, and so on and so on. The basidiomycetes have and produce spores, and they form mycorrhizal relationships and are plant parasites and or decomposers of wood. The phylum is defined by the club-like structure called the basidium. So this right here, where my mouse is at, is not the club-like structure. It's what's under it. So this right here, the club-like structures right here, is what gives them their definition. The basidium. So this is the basidium right here. This is not the basidium. So these are different types of basidiomycetes, such as puffballs, shelf fungi, and maiden veal fungus. The maiden veal fungus is my favorite because it looks fancy, like pretty cool, right? That was a voice crack. So plasmogamy. So not plasmogamy, sorry. We'll be looking into the life cycle of a basidiomycete. Right? I said that right? Basidiomycetes, yeah. So, starting with right here, haploid mycelia, their mating types match, and once they match, they will undergo plasmogamy, which means they fuse their cytoplasm, and here we have the awkward stage of one nucleus from the mating type negative and one nucleus from the mating type positive. This mycelium will grow and grow and grow into a basidiocarp. It is still in the it is still in the dikaryotic stage or heterokaryotic stage, meaning that the whole basidiocarp or the whole mushroom structure has not underwent karyogamy yet. Karyogamy happens in the basidium. So once karyogamy takes place in the basidium, which is right in the fins or the gills of the mushroom, it will undergo meiosis, making 4 haploid nuclei, and it will undergo mitosis, creating spores. And those spores would be launched out away from the basidium or spit out like that, like throwing up, I guess. And the cycle is repeated. Basidiomycetes can produce mushrooms within a few hours. So, ring fairies, fairy rings, is that what they call them? They're really not that special. They're just a natural phenomenon because their mating types are found here. Fungi are great decomposers of organic material. They decompose cellulose and lignin. Cellulose is the polysaccharide in plants. Lignin is the main component of wood. And they perform essential recycling of chemical elements from organic to inorganic forms and vice versa. Fungi is mutualists. Fungi form mutualistic relationships with plants, algae, cyanobacteria, and animals. 85% of plants use mycorrhizal formations or relationships. Agriculture is greatly enhanced by the presence of fungus. So here we'll be looking at this study right here. So there was a study, data by A.E. Arnold et al. Okay. So here we have the cocoa plant. This cocoa plant had a control and a experimental. So an endophyte not present and a pathogen present. Wait, hold on. Let me look at this real quick. Leaf mortality percent. So a pathogen, here we have the pathogen present right here. Look at this. This is the pathogen present. And the endophyte is a type of mushroom that the cocoa plant has. So here in this plant, let's call this plant A, the plant did not have the endophyte fungus, but was exposed to the pathogen. The, the plant A suffered greatly because the leaf mortality was high, causing a disease. Now, a plant with both the fungus and the pathogen, it did not lose much of its leaves due to the disease. And here is more proof right here. No fungus, pathogen present, higher leaf mortality rate. Fungus present, pathogen present, lower leaf mortality rate. Fungus and animal mutualisms. Some fungi can share digestive services with animals. Leafcutter ants will purposely bring undigested leaves to their colonies. And these colonies would have hyphae or mycelium or funguses to break down these leaves that the ants have laid out for them. This fungus would create sugars, minerals, and edible food for the plants that was once a leaf that the ants could not eat. Lichens are a symbiotic association between photosynthetic microorganism and a fungus. The microorganism can be a green algae or a cyanobacteria, and the fungus is usually an ascomycete. The algae provides carbon compounds such as organic compounds such as sugars, while cyanobacteria also provide organic nitrogen and fungi provide the environmental for growth. The fungus also provides protection to these microorganisms. Hold on. But these produce asexually. So here we have the ascocarp of a fungus, the fruiting body, and here we have an algae cell wrapped around in fungal hyphae. So you would say that the hyphae are around and are wrapped around. Well, the algae cell is wrapped around hyphae. Fungi are also parasites, as we mentioned in the beginning. About 30% of known fungal species are parasites or pathogens, mostly on or in plants. 10% to 50% of the world's fruit harvest is lost to fungi each year. So 40,000 people died from ergots, which is a type of funguses that grow on rye, and they produce a toxic compound. There's also funguses that attack chestnut trees, and animals are less susceptible to fungal infections. So, us humans, we don't experience so much parasitic fungi. Well, I worded that wrong. Us humans and animals are not greatly affected by parasitic fungi. Plants are more the victim. And here is a study case where a bunch of frogs in California just simply got a fungal infection because the amphibians have sensitive skin because they absorb water and breathe through their skin. B. dendrobacteris killed a crap ton of these frogs, and it almost drove them to extinction, and scientists had to treat them with fungicides. The term for a fungal infection in animals is called mycosis. Ringworm and athlete's foot are examples of human mycoses. Systemic mycosis spread through the body, while this type of fungus called a a coccidio, okay, I'm gonna have a little trouble. Coccidioidiomycosis produces tuberculosis symptoms. Some mycoses are opportunistic. Candida albicans, which causes yeast infections, which is commonly found in women. Although there is practical uses of fungi, humans eat many fungi and use others to make cheeses, alcoholic beverages, and bread. Yeasts make alcohol. If you have taken Bio 1, you would understand that yeasts go through, what was it called? Alcohol fermentation, where you learn about acetaldehyde accepting an electron, becoming alcohol. And some fungi are used to produce antibiotics for the treatment for bacterial infections. Penicillin, which is made by Ascomycetes, penicillin was an antibiotic and it changed the world because it helped us fight bacteria such as the staph infection and, well, now, well, a weaker version of staph, I guess. Now there's MRSA. Well, antibiotics changed the battleground for humans versus bacteria. So here is a sum up. So chytrids have flagellated spores. Mucoromycota have resistant sporangium as a sexual stage. Ascomycota, sexual spores, ascospores, born internally in sacs called asci. Vast numbers of asexual spores called conidia, asexual spores called conidia produced, and the Basidiomycota have elaborate fruiting body, basidiocarp, containing many basidia that produce basidiosexual spores.